we'll finish with eukaryotic gene expression regulation. And we left it on Tuesday at transcriptional regulation. We didn't even start. We talked about pre-transcriptional regulation. So for transcriptional regulation in eukaryotes, it's not much different from what we covered for prokaryotes, right? And in fact, transcriptional regulation is somewhere in the middle in the previous slide because the mechanisms used in both domains are fairly similar. So quick reminder, on top here we have the typical structure of a eukaryotic gene where broken arrow or bent arrow indicates the TSS. Notice how the ATG is somewhere after that TSS because TSS and ATG are not the same thing. ATG, we call it ATG in DNA, but in RNA it will be AUG, the start codon, right? Then these blue boxes here are the exons. The gray boxes in between are the introns. We'll talk about splicing again in a few minutes. Now, prior to that TSS, there is the core promoter that in eukaryotes is any collection of these consensus sequences that can happen in any combination, any order. So we have TATA boxes, CAT boxes, GC-rich boxes. All of these are classes of consensus sequences that you can find in eukaryotic core promoters. This is where the transcriptional initiation complex will assemble. That's the TF2 family of proteins, and they will recruit RNA polymerase. Now, upstream of the core promoter, there is what we normally call a regulatory promoter, a proximal regulatory promoter. And this is, by convention, typically you will find these proximal regulatory promoters all the way up to about 1.5 kilobases upstream of the TSS. So up to about 1,500 base pairs upstream of the TSS, you might still find binding sequences for proteins that will regulate the expression of a gene, and hence the name regulatory promoter, right? So the way these regulatory promoters work is not much different from what we saw in bacteria. In the core promoter, you'll have the assembly of the initiation complex, those TF2 family of proteins that I mentioned before, and RNA polymerase. But then there's going to be these transcriptional activators that will induce that assembly, and also some transcriptional inhibitors that may inhibit it. So at the end of the day, what happens is regulatory promoters will control the expression of their, what we call target genes, meaning the genes that they regulate, based on some sort of balance between the activators and repressors present in those regulatory promoters. So you may have a regulatory promoter that is solely occupied by activators. In that case, the expression of that gene will go up. You may have a regulatory promoter solely occupied by repressors. In that case, the expression of that gene may be turned off completely. Or you may have anything in between, right? So some combination of activators and repressors that will make that expression somewhere in the middle, right? And all of these, like in bacteria, is a very dynamic process too. These transcriptional activators and repressors are binding to DNA through target sequences that they recognize through their DNA binding domains, right? But those bindings are not covalent. They're not chemically bound to these DNA sequences. They're just interacting with them. And what that means is that these proteins that are interacting with their target sequences in DNA can come onto DNA and bind to it, or come off from DNA and leave, right? And that is often regulated by allosteric domains, just like in bacteria. So these transcriptional activators or repressors may be made to bind DNA or come off of DNA based on interactions they have with other molecules in cells, like inducers, other proteins that bind to them, and so forth. Okay? And last but not least, there is the transactivation or transrepression domains. Now it makes a little more sense to go a little deeper into these. In eukaryotes, often these transactivation and transrepressor domains, unlike in prokaryotes, they often operate indirectly. What I mean by that is this. Let's say an activator, a transcriptional activator, may come to the regulatory promoter of a gene, bind to DNA, and it may not directly recruit TF2 family proteins or RNA polymerase to the core promoter. Instead, what it does is it recruits some of those histone modifying enzymes that will, for instance, acetylate histones in the region and therefore loosen up DNA. And that is what indirectly facilitates the assembly of the initiation complex at the core promoter. That happens a lot in eukaryotes. So unlike prokaryotes, where that activation or inhibition of transcription is more direct, you have, for instance, those repressors blocking the passage of RNA polymerase, physically just stopping it, or activators that interact with the sigma factor and RNA polymerase to bring them to the core promoter. In eukaryotes, there is that extra layer or extra mechanism for regulation that involves chromatin modifications. So the activator or the repressor comes to the regulatory promoter, modifies chromatin locally, loosening it or tightening it, and therefore making it easier or harder for the initiation complex to assemble and initiate transcription. Any questions, anyone? So if that sunk well, you'll probably appreciate something that is kind of unique of eukaryotes, kind of, and that is the concept of touch and go regulation of gene expression. What that means is you can have either activators or repressors that bind to DNA briefly, modify chromatin, and then they leave. So they may act to regulate the expression of a target gene, but they don't need to be present while they're doing that. That happens quite a bit in eukaryotes as well. So you can have a repressor that comes, tightens chromatin locally for this particular gene, and then it goes away. And those histone modifications, as we saw the other day, are reversible but fairly stable. Unless the cell commits to taking those histone modifications away, they'll stay, right? So if those histone modifications were such that the binding to DNA is tighter now, then that gene may stay repressed even though the repressor left already. Okay? Did that make sense? Cool. All right, so the other feature that is rather unique to eukaryotes, this you don't see in prokaryotes, is this idea that collections of binding sites for transcriptional regulators, be it inhibitors, activators, or both, don't have to be immediately upstream of the core promoter. I mentioned that any area within 1,500 base pairs upstream of the core promoter we normally call the proximal regulatory promoter. But sometimes these binding sites for transcriptional regulators can happen far away. Not only far away upstream of the core promoter, as shown in this diagram here, where you have the core promoter here, but then these binding sites are much further away. Let's say that they're located, 10,000 kb away upstream of the core promoter. So not only can they happen much further away upstream, they can also happen downstream, like shown here. So we go in this direction, and 50 kilobases downstream of that core promoter, there is a bunch of binding sites for transcriptional regulators that have the ability to control the expression of this gene here, of this core promoter here. How does that happen? We touched on this briefly when we talked about transcription, because of DNA looping. DNA at this scale behaves like a loose string. So even though you have the proteins binding to DNA far away from the core promoter, when you think about the number of base pairs away that those binding sites are from the core promoter, because DNA can loop on itself, it can bring those proteins close to the core promoter again. The one? KB is kilo bases. So that's a shorthand that we use for 1,000 base pairs in DNA. Yeah. Did that make sense? These collections of binding sites that can operate on a target gene from a distance, we call, you guys remember? Enhancers. Enhancers. Now, one thing that it's a little misleading about that name, historically, the first sequences that control gene expression at a distance happen to enhance the expression of their target genes. And we call those things enhancers. Now we know a little better, and now we know that there are enhancers that can also repress the expression of their target genes. So you can have enhancers that are far away from their target gene from a DNA sequence perspective, but because of DNA looping, the proteins, i.e. transcriptional repressors bound to them, can get really close physically to the core promoter and inhibit the expression of those genes. So even though we call them enhancers, they don't always need to enhance. Sometimes they can also repress. To the point that there are enhancers that all they do is repress, and we call those? Close. Silencers. Silencers. Again, fairly self-explanatory term. They just silence the expression of certain genes. And last but not least, there are sequences that, again, operate at a distance that we call insulators. And what they do is they serve to kind of establish compartments in DNA such that whatever is going on in one section of DNA won't affect the neighbor section. How these insulators work is by creating these links to nuclear membrane, blah, blah, blah. There's a whole series of mechanisms. But at the end of the day, what they do is that they insulate, they isolate certain regions of DNA from whatever else is going nearby. Okay? All right. So that's all I have to say about transcriptional regulation. A couple more layers, right? But at the end of the day, the essence is the same. Proteins that bind to DNA through specific target sequences that they recognize biochemically. These proteins can bind DNA or not, depending on their shape, and that shape is often controlled by molecules that interact with them through allosteric sites. So you can have transcriptional regulators, be it activators or repressors, in an open and active state or a closed and turned off state, if you will. And they regulate transcription by facilitating or inhibiting the binding of the transcriptional machinery. Now, in prokaryotes, it's direct. They will bring RNA polymerase or block it. In eukaryotes, it can get a little more complicated in that these things can act indirectly by modifying chromatin, and in some cases even leaving. Okay? Cool. So, brief summary, transcriptional regulation. In prokaryotes, pretty simple. Core promoter and proteins that bind around it, facilitating or not the assembly of the holoenzyme. In eukaryotes, they can act right upstream of the core promoter, like in prokaryotes, or at a distance in either direction. And that is made possible by DNA looping. Okay, so now let's move on to post-transcriptional regulation. And I bolded regulation here, and I faded 5' capping and 3' polyadenylation for a reason. It turns out that all mRNAs go through 5' capping. All mRNAs go through 3' polyadenylation in eukaryotes. And cells haven't really found wiggle room in those two processes for regulation, right? So these are things that are done to all genes. So when you do something to all genes, that's something that cells can't really use to modify the levels of expression of a gene, right? There isn't anything like a little 5' capping or a little 3' polyadenylation. It's there or not. mRNA is needed to survive in the cytoplasm and to be translated. So it's an all or none. There's no room for for regulation.
Is the reason why bacteria doesn't do post-transcriptional regulation is because translation and transcription can happen at the same time?
Correct. Yes. So there's two arguments to that. So one is it's a physical consequence of the fact that there is no time between transcription and translation. So why do post-transcription translation when your mRNA is going to be translated right away? So that is the mechanistic explanation. Then there is what is the adaptive value of that evolutionarily? Why would bacteria do cold transcriptional translation? And the answer to that is because it makes things quicker. And if you're a simple organism that all it wants to do is grow and reproduce, that works, right? Economy, speed, that works. It's only with multicellular organisms that want to do very complex things that you need to add all these extra layers of regulation. So even though these three processes, all three are post-transcriptional modifications of RNA, the only one that is really regulated, the only one that allows cells to introduce some level of regulation is alternative splicing. And we'll discuss it next. So we mentioned this briefly when we talked about splicing itself. Most, not all, most eukaryotic RNAs for protein coding genes are expressed as pre-mRNAs that include sequences that will be present in the mature mRNA that is eventually translated, and then some sequences that need to be taken out, and that's the process of splicing. When we say that a given pre-mRNA undergoes alternative splicing, what we mean is that the cell will splice the exons one way, sometimes a different way other times. Okay?
Would we do it at the same time either way?
Excellent question. So, yes to all of the above. In a way, there will be cells that always do it one way, cells that do it always the other way, cells that do both, but depending on conditions, they may do more of the one or the other. Right? So, all of those scenarios happen. Sometimes it is critical for the identity of a cell to always do one, not the other, and they'll do it. Other times, these different versions of the splice variant will depend on what conditions that cell is in, and they may choose to do one or the other. And we'll discuss how that happens. All right, so alternative splicing is just that. The same pre-mRNA can be spliced differently depending on different conditions. Let's say, for instance, that we have these two cell types, cell type A and cell type B. This kind of goes along with your idea that maybe cells do it one way, other cells do it another, though it could be the same cell type A, under condition 1 and condition 2, okay? But in this example, it's two different cell types, A and B. It turns out that, let's say, so these two cell types splice as shown in the diagram. So cell type A will splice exons 1 and 3 together, whereas cell type B will include exon 2. Right? So that's the scenario. How this happens is, in cell type A, the spliceosome, the machine that does the splicing, recognizes the donor site present at the 5' end of exon 2 as a donor site and the 3' acceptor site of this intron. Whereas in cell type B, that donor site is not seen by cell type B, and instead the one that is seen is a donor site that happens between exon 2 and intron 1. Did that make sense? So splicing, it depends at the end of the day on what the spliceosome, the machine that does the splicing, sees in these pre-mRNA molecules. So cell type A is seeing the upstream donor site and the downstream acceptor site, and cell type B seeing the downstream donor site and the same acceptor site. So both cell type A and B see the 3' acceptor site, but Cell type A sees the first 5' donor site, and cell type B sees the second. When I say a spliceosome sees these sequences, what I really mean is that the spliceosome will contain different SN RNAs. You guys remember SN stands for small nuclear RNAs, and these are one of the many non-coding RNAs that get loaded onto the spliceosome, and the spliceosome uses to scan for donor sites and acceptor sites, right? So at the end of the day, what it comes down to, whether a spliceosome sees a given donor site or an acceptor site, is whether or not they can read, quote unquote, these sequences in the pre-mRNA molecule. So my question to you is, how can different cells see a donor acceptor site or not? How could, let's say, this first donor site be invisible to cell type B?
Acceptor site.
Donor site or acceptor site. How do spliceosomes look for donor and acceptor sites? What do they use to look for these things? It is the snRNAs. These snRNAs will find donor and acceptor sites based on base complementarity. So just like proteins that can read specific words in DNA, these snRNAs can also read specific words in the pre-mRNA molecule. And they will go, and through base pairing, they will tell the rest of the spliceosome that they're contained in, hey, we found a donor or acceptor site, right? Spliceosomes are big machines with different parts to them, and there are parts of a spliceosome that are in charge of looking and finding donor sites, and parts of the spliceosome that are in charge of looking and finding acceptor sites. And when the spliceosome feels that it found both, that's when the actual cutting and pasting reaction takes place. The details of that we discussed when we talked about splicing during transcription translation in eukaryotes, right? The whole cutting of the 5' end, looping, branching with the branch point, formation of the lariat, and all that. Right? But from a regulatory perspective, different cells will splice their pre-mRNAs differently by simply seeing or not different combinations of donor sites and acceptor sites. So one major way in which a cell may not see a given donor sequence in this example is by not expressing the one snRNA that would allow them to see that donor site in a pre-mRNA. They express the other. And I think that's expressed here. So option 1, cells can express different snRNAs and may thus be able or unable to base pair with a given splicing sequence. But that's not the only option. The second option is a lot less intuitive. So I'd like to ask you all first this question. Here you see a cartoon where presumably we're going through the different steps of protein coding gene expression in eukaryotes, right? We see transcription on DNA in the nucleus. There is the pre-mRNA capping at the 5' end, then polyadenylation and pre-mRNA splicing. When the transcript has been properly spliced and properly polyadenylated and capped, it can be exported. And then once in the cytoplasm, there is a number of steps that will control initiation of translation, and among others, mechanisms to control for RNA stability. And then translation takes place, and you have a protein. My question to you is, what's wrong about this image?
You don't see the introns?
You don't see the introns? So the introns are here, these yellow. Oh, afterwards. Yeah, but so in terms of this cartoon, there's nothing wrong. Oh, you mean the lariats? That we don't see the lariats? Is that what you meant? Okay, fair enough. So we don't see the lariats. So those are, that was white. Wait. There you go. So we should add the lariats here. Okay, cool.
RNA is usually not that straight, so
Not die straight. So what were you expecting to see in an RNA molecule?
Loops, because that's how it stops the polymerase.
Excellent. So we should add some loops here, right? Oh, again. So that's closer to reality. Any RNA molecule will likely engage in some intramolecular base pairing and form these famous hairpins. Cool. What else? Here's the one thing that is the least intuitive of all, and I would be surprised if any of you knew this, but that's why you're here. It turns out that if we were to look in cells, RNA is rarely floating around naked. It's from the moment that it begins to be transcribed, is coated with a whole bunch of small proteins called RNA binding proteins, or RBPs. Right? And the reason why cells code their RNAs with these proteins is, as usual, twofold. Protection is a big part, protection of its own RNA molecules so that nothing destroys them, but also regulation, right? So just when you think that RNAs are floating around as RNA molecules in cells, in eukaryotic cells, think again, they're normally coded by a bunch of RBPs. So now knowing that, we may appreciate the second option for alternative splicing, and that is option number 2. What happens sometimes is that those splicing donor or acceptor sequences may be hidden from view, may be hidden from the spliceosome by RNA binding proteins or those secondary structures that were mentioned before. If those hairpins that form happen to involve a donor or acceptor site, the spliceosome won't see them anymore. Right? So now, of course, the question becomes, well, what's going on in cell types A and B? Well, it could be that cell type A produces an RBP that binds the second donor sequence, so the only one that it sees is the first. Alternatively, it could be the opposite, that cell type B produces I'm going to draw it here, produces an RBP that happens to bind RNA where the first donor sequence is, so it sees only the second. Or it could be that either cell type the RNA in either cell type happens to form a secondary structure right where those donor sites are. Now, why a given RNA molecule may form a hairpin in one donor site, not the other, and vice versa in the other cell type, well, that again depends on what else is going on with that RNA molecule. So, for instance, the presence of an RNA binding protein here might prevent the formation of a hairpin right after it. Because for these hairpins to form, they need to have full access to RNA sequence, right? So everything is kind of interconnected, but the point that I wanted to make here is that there is two things that come into play when a spliceosome recognizes and uses a donor or acceptor site for splicing. Whether the spliceosome has the ability to see those donor and acceptor sequences, that depends on what snRNAs they carry, and whether or not those donor and acceptor sites are visible. Or, alternatively, they may be hidden in secondary structure or by proteins that are binding those regions in RNA, RBPs that are binding those regions. So now it all comes down to what snRNAs, cell type A and B express, what RBPs, cell type A and B express, and so forth. And not only that, back to what we were discussing before, sometimes the same cell, depending on external conditions, can express one set of snRNAs or another. They can express one set of RBPs or another. All of those are decisions that the cell will make through a series of decision steps based on information they have from the outside and the inside. Not much different from what we saw with the lac operant. With the lac operant, super simplistic, but there were two decisions to be made. Should I express the operand or not? That depending on the presence of lactose. And then there was a decision to be made about the levels of expression. That makes sense? Yeah. Okay. So, alternative splicing always alters the sequence of proteins that are coded by a given gene. True or false? Think about it for a few seconds, talk to your neighbors. The statement is that alternative splicing will produce different proteins. Is that true or false? So why is it true? Why will these alternative spliceoforms, by the way, we call them that in technical jargon, so different variants of an RNA after splicing are different spliceoforms. Why will different spliceoforms give rise to different proteins?
Because it's only picking and choosing which exons are inside the DNA.
Okay, and that changes the sequence of nucleotides, and that will change the sequence of amino acids, right? Now, I take it that either everybody's super shy or many of you thought that this statement is false. So now I'd like to hear from you. Why would it be false? Why could it be that these different spliceoforms express the same protein?
There's multiple amino acid combinations to produce each protein, right?
Mm-hmm.
So if the splicing just creates a different version or different option for the
Oh, I see.
protein.
So what you're saying is the protein is the same even though it uses a different combination of amino acids.
Yeah.
Because different amino acids can lead to the same structure. And that's ultimately what matters for a protein. I did not see that one coming. It's a valid argument, but it would still, yeah, I see. I should have said sequence of amino acids. That's my bad. That changes everything, right? So I see what you're saying. What you're saying is you can have the same protein with different amino acid sequences. Let's make that statement more general. Will these different spliceoforms always give rise to different amino acid sequences? Yes? Yeah? Yeah. No, and here's why not. It all depends. So the alternative splicing will give you different combinations of exons, right? But it all depends on where the AUG and the stop codon are. And to illustrate that point, let me say that in this particular example, the AUG is here and the stop codon is here. In that case, the AUG and the stop codon in both isoforms and everything in between will code for the same sequence of amino acids. So in this case, what alternative splicing changed, and this happens a lot, by the way, is UTR sequence, untranslated region sequences. Things that happen before the AUG or after the AUG or after the stop codon, they don't affect the sequence of amino acids coded by that particular mRNA, but it may affect other aspects of the life of that mRNA. It may affect their stability. It may affect where that mRNA ends up within a cell. It may affect whether or not its translation is regulated by certain processes or not. All of these things, mRNAs, or the information for all these things, mRNA encodes in 3' UTR and 5' UTR. mRNA save the sequence between the AUG and the stop for just the amino acid sequence. That's all that chunk of sequence encodes, right? All of this extra information that says to a cell when, where, and how much an mRNA should be translated is in the 3' and 5' UTR.
So the start and stop codons, that's the protein.
That's right. Yeah. AUG and stop delimit the protein coding sequence of an mRNA. Before that, we have 5', shake it to them, too much coffee, 5' UTR. And after that, we have 3' UTR. We've talked about this when we talked about gene structures, something to remember. So not all nucleotides in an mRNA will be translated into amino acids. There is sequence that is not translated before the AUG, sequence that is not translated after the AUG, after the stop. And this applies to both eukaryotes and prokaryotes. And both eukaryotes and prokaryotes use information encoded in those UTRs to do things to those mRNAs. Yeah.
So splicing in non-protein coding sequences, not that it's not important, but it doesn't affect like the function of the protein?
Exactly. So it may not affect the biochemical function of the protein, but by affecting its levels, by affecting where that protein is expressed, by affecting when that protein is expressed, its function within the life of that cell may be regulated. The same enzyme expressed at very low levels may do something inside of the cell, right? But it may not affect certain processes. The same enzyme expressed 10 times at levels 10 times higher, now all of a sudden it may be affecting the metabolism of that cell differently. Did that make sense?
Because of the splicing in the non-coding.
For in the non-coding region. So for instance, you splice the 3' UTR differently. In this case, for instance, the purple exon is included or not. And if that purple exon regulated the stability of that mRNA, making it more stable, this mRNA down here is less stable, will be shorter lived, and not as much protein is produced from it. That guy up there is longer lived, produces much more of the same protein. So even though the function of the protein is the same, from a cell perspective, upper mRNA produces more of that function than the lower. And speaking of that, let's talk about another post-transcriptional regulation mechanism that we didn't mention before, but now is the time to talk about this, and that is the regulation of mRNA stability by miRNAs and siRNAs. miRNA stands for microRNAs, and siRNAs stands for either silencing RNAs, or sometimes people call them short interfering RNAs. Doesn't matter, siRNAs, okay? So the way this works, I'm not going to go into the nitty-gritty details. You can see the slide later, and there's gazillion, well, not a gazillion, but there are videos out there that show this in detail, and the details don't matter that much. But the bottom line is, miRNAs are short. Let's start with from the bottom, right? So miRNAs, microRNAs, are short RNA sequences, non-coding RNA sequences that get loaded onto this big protein called the RISC complex. Right? So it's not one protein, it's a bunch of them. And these miRNAs that get loaded onto the RISC complex are used to probe the cytoplasm for any mRNAs that may have partial base complementarity to them. If that sounds familiar, it's because we just discussed something very similar with spliceosomes. Spliceosomes load an snRNA and they probe for donor and acceptor sites. The RISC complex does something similar. It loads an miRNA and probes the cytoplasm for messenger RNAs that may show some partial complementarity to them. If they find them, and there is imperfect base pairing, meaning not all bases need to be complementary to the bases in the mRNA, but some partial complementarity, the RISC complex stays bound to that mRNA, and more often than not, what it will do is prevent its translation. So mRNA is there, but it's not being translated because it has this huge thing, the RISC complex, attached to it. And our ribosomes can't work with that mRNA anymore. Okay? Now, I started from the bottom. I started the story from the end. How this miRNA molecule, this short RNA molecule, got loaded onto the RISC. Where do the miRNAs come from? They come from genes that are encoded in DNA and that have this peculiar feature. They have what are called inverted repeats. They evolved to have these inverted repeats. What happens is when these sequences get transcribed into RNAs, they will form these famous hairpin loops. Why? Because it's a stretch of sequence that is complementary to a stretch of sequence that is in the inverse orientation a little later in the same molecule. So the molecule will fall on itself and form one of these stem loops or hairpins. When that happens, there is another complex, an enzymatic complex inside of cells that will cleave the hairpin first. Then the hairpin gets processed further, so the loop gets chopped up, leaving this short-stranded, double-stranded RNA molecule. That was a tongue twister. I meant a short double-stranded RNA molecule. And when I say short, I mean somewhere in the vicinity of 20 base pairs. And one of those serves as the microRNA that gets loaded onto RISC. So one of them gets tossed out, the other one gets loaded onto RISC. Okay? And from that point onwards, that RISC complex loaded with that microRNA will patrol the cytoplasm looking for target mRNAs. And if it finds them, it will inhibit their translation. So this is a mechanism that is after transcription of an mRNA that regulates the expression of the protein encoded in that mRNA, because that mRNA is there but cannot be translated. Okay? Cool. siRNA inhibition is very similar. I have them here side by side so that you can compare later, except that the base pairing of siRNAs is perfect to their target sequence. Right? And when the base pairing of the siRNA is perfect to the target mRNA, RISC will not just inhibit translation. It will actually go ahead and cleave the mRNA. So this mRNA gets cut. Moreover, when you have inhibition by siRNA, it's usually not one siRNA that maps to a given mRNA, it's several of them. So these mRNAs get literally chopped up into smaller pieces. The end of the story is the same except for that detail that siRNAs show base pairing that is perfect, 100% base pairing, versus the imperfect base pairing of mRNAs. So that's one aspect in which siRNAs differ from miRNAs. The other aspect is where these siRNAs come from. Unlike miRNAs, these siRNAs are rarely coded as separate entities in DNA, right? They are produced by chopping up much longer double-stranded RNA molecules. I mentioned that for microRNAs, there is these short genes that get transcribed, and because of their inherent structure, they will form the hairpin. That hairpin is recognized by the enzymes that will chop it out, then process the loop, take the loop out, and separate the two strands of those short double-stranded RNA molecules. siRNAs are processed a little different. They start from longer double-stranded RNA molecules, they get chopped up into small pieces, and then each of them gets separated such that one of the halves is tossed out, the other one gets loaded onto RISC. Did everybody get that? One more time. So miRNAs come from genes encoded in DNA. Those genes are encoded in such manner that when they get transcribed, they will form a hairpin, and the hairpin gets processed into a microRNA loaded onto a RISC. siRNAs, on the other hand, are produced by chopping up much longer double-stranded RNA molecules into small pieces, and each of those small pieces can be separated. One half gets tossed out, the other half gets loaded onto a RISC.
So they're pretty much just like double-stranded RNAs. Where these
Double-stranded RNAs, right?
versus where miRNAs are part of like DNA.
DNA, right? So miRNAs are encoded in the DNA. siRNAs come from double-stranded RNA. Where is that double-stranded RNA from? Who uses double-stranded RNA? Close. Viruses. Viruses. There are many, many, many viruses that their genetic material is encoded in double-stranded RNA. And when they infect a cell, they will introduce double-stranded RNA into the cell. So the siRNA pathway evolved as a defense mechanism against viruses. A virus infects, the cell will chop it up into small pieces. It will use those pieces to probe for other viral molecules that may have infected the cell. And if it finds them, or rather the RNAs that are produced from those viruses, it will destroy them. Or if it finds the virus itself, it may destroy it as well, right? So it's a mechanism, a defense mechanism to ensure that no viral proteins are produced within an infected cell, because the cell will chop up the viral sequence into small pieces, and many of them will obviously map to genetic information that the virus uses to produce viral proteins. Those sequences will eventually make it into an mRNA molecule, and those are the ones that the risk will find and destroy.
Kind of off topic, but is this kind of how like vaccinations work?
Yes and no. So the analogy is in that there is a certain memory in the organism of having seen that virus before. On a cellular level, siRNAs keep a memory of a virus by chopping it up and then creating many of these risks and patrolling. Should another virus of the same kind show up, they'll attack it. With vaccination, there is a whole immune system involved, right? So there is an immunization with a tempered version of that virus. The immune system learns, oh, this is a foreign entity. We should learn to detect it, and if we see it again, we'll attack it. And that's how vaccines work. If you believe in vaccines. I'm kidding.
Sorry, so going over it again. mRNA is encoded in DNA.
That's right. miRNAs, there are genes that we call MIR genes, and they're genes with a promoter, regulatory promoter, they're non-coding, so they won't produce proteins, but they're transcribed like so many other non-coding RNAs. In fact, I know it sounds like an eternity ago, but when we talked about non-coding RNAs, we listed these guys. We mentioned PIWI RNAs, miRNAs, siRNAs. Back then, it was like some, nebulous concept. But now it's making sense. So these genes exist in DNA, and they're transcribed, and their transcription is regulated like any other gene. The difference is that instead of giving rise to a transcript that will eventually make it into an mRNA that gets translated into a protein, these guys get processed in such a way that they generate those short double-stranded RNAs that risk will toss one, keep the other, and start patrolling.
So for the siRNAs, it says it can also come from long hairpins. Is that more or less common than coming from a virus?
Way less common. And it's something that evolution did much later. So evolution created this system for defense against viral infections. And once this system was in place, some cells with some genes, or some species, I guess, with some genes said, hey, if we can quickly destroy any mRNA using siRNAs, can't we make siRNAs against our own mRNAs? It's a very rare phenomenon, but there will be cells that will make double-stranded versions of their own genes when they want to turn off their expression quickly. So it's almost like I've been expressing this gene forever, but now I need to not only not express it anymore, I need to turn it off now. So what they'll do is they'll transcribe that gene from both ends. Yes, they use promoters on both ends of that gene. When those two long transcripts find one another in the nucleus, and they will, they form a double-stranded RNA, and the cell, the rest of the cell, poor little cell, thinks, oh, virus. It triggers the whole siRNA pathway, and next thing you know, it's the cell's own mRNA is being quickly destroyed. And that's how a cell can turn off expression of its own genes quickly.
So would that be considered as their own sort of like promoter, like activator?
In a way, but everything that I just described, we've elevated the conversation, guys. This is like 400 level graduate school level discussion. I mean it. But it happens. It's rare though. It's very rare. All right. So I had a slide kind of priming the question here. mRNAs typically show imperfect base pairing, whereas siRNAs have perfect base pairing. Why? In a way, we discussed all of this because of where they come from. So miRNAs come from genes in DNA that evolve separately from all of their target genes. So throughout evolutionary timescales, I look at you because you were asking this question. Throughout evolutionary timescales, there's going to be some divergence in sequence, right? So the microRNA gene that gives rise to a given microRNA may suffer a bunch of mutations here and there, and so will some of the target genes. So there's going to be some divergence in sequence, and that is why there is no perfect matching between the mRNA, miRNA, and its targets, right? Throughout evolution, there's been some divergence. With siRNAs, it's all within the lifetime of a cell. So the cell sees that double-stranded RNA, mostly because of viral infections, sometimes because of this weird double transcription or transcription in both directions. And within its own lifetime, no evolutionary timescales, it will create the siRNAs to destroy those sequences. Okay? So why miRNAs don't have perfect matching to their target sequences is because we've introduced millions of years of evolution between the microRNA gene and its targets. Okay? And one feature about microRNA genes is since they don't require perfect matching, they can target several different genes. So there will be several different mRNAs that may have sequences that are close enough for microRNA binding and therefore risk silencing. The difference is that they're not destroyed. Those mRNA molecules are not destroyed. They're just prevented from being translated for a while. Okay? Cool. So we're almost there. In eukaryotes, there are mechanisms for translational regulation. I'm going to go quickly through this because these slides are pretty self-explanatory, but we mentioned briefly that in eukaryotes, the small ribosomal subunit will bind to a bunch of proteins that are bound simultaneously to the 5' cap and the poly-A tail. So that's a way that the small ribosomal subunit knows that an mRNA in eukaryotes is whole and it's intact. There's nothing missing, right? So it binds to them, and what happens is it will then start the scanning for that AUG. This is what we call the initiation of translation in eukaryotes, right? It's everything that happens before that ribosome finds the AUG and actually starts translation. This process, the initiation of translation, can be regulated. And the way it is regulated is as follows. There is this, it's actually it shows there as if it were only one protein, but it's a family of them that we call EIF4BP, right? Details don't matter that much, and you probably don't need to remember this name in particular. If it shows up in the exam, you can ask me and I'll tell you. But these proteins, what they do is they block the ribosome from initiating that scanning process. So everything is in place, except that EIF4BP is stopping things for now. The mRNA is intact, the small ribosomal subunit is bound to that intact RNA and it's ready to start scanning, but that protein will keep it from doing it. Until something tells the cell, hey, we should translate this thing now. So there are a series of chemical reactions that lead to the phosphorylation of EIF4BP. Phosphorylation, as we will see in a few seconds, is a mechanism by which phosphate groups are chemically attached to certain amino acids in certain proteins. In this case, there's a whole bunch of phosphate groups attached to certain amino acids in EIF4BP. And when that happens, EIF4BP can no longer bind the mRNA, falls off from it, and now small ribosomal subunit is ready to start translating that mRNA. So we call this translational regulation because it is a regulatory step at which the cell decides if an mRNA that is ready to be translated is or is not. Yeah?
Is there like a specific kinase to add phosphate groups?
Yes, you said kinase is kinase. Okay. That's how you put kinase. Kinase are the enzymes. It's a very generic name, but kinase are enzymes that add phosphate groups to target proteins. And yes, there is a specific kinase for EIF4BP, yes. And that kinase or those kinases are the ones that sort of integrate information from within and outside of the cell, I suppose, that will tell them, oh, now you should go phosphorylate for EIF4BP or not, right? So things that will turn on or off those kinases are the ones that will phosphorylate or not BP. And
then later on, can that be phosphorylated and added back to it?
yes. So the question is, are these phosphorylation reversible? And the answer is absolutely yes. So just like there are kinases that add the phosphate, there are phosphatases that take them out, right? And make that EIF4BP able to bind mRNAs again. Another thing is not all mRNAs are sensitive to regulation by EIF4BP. Some are not. And it all comes down to whether or not 4BP sees specific sequences in the 5' UTR of those mRNAs. So again, alternative splicing that did not involve coding sequence but involved 5' UTR could, for instance, make an mRNA regulatable by 4BP or not. So one cell type may regulate the translation of a gene, a different cell type that expresses a different spliceoform with a different 5' UTR that does not have a target sequence for BP will be completely insensitive to BP regulation. Yeah? I'm going to breeze through the last slides related to post-transcriptional regulation because we have talked about some of these things before, and they're all, again, pretty intuitive. There are several mechanisms by which, after a protein has been translated, the cell can decide if that protein will be fully active or not. Many proteins are not fully active right out of the ribosome. They still need some work done to them. And what is done to them comes in a number of flavors. One is proteolytic activation. So that's a mouthful, but proteolytic means that a protein has been broken, right? And ironically, when you go and chop up these proteins or take out certain regions of these proteins, one intuitively might think, oh, you're breaking it. It's quite the opposite, you're activating it. So these proteins exist in preforms that are inactive, and then the chopping up of their sequence, the taking away of some amino acids will activate them. The example on the screen is proinsulin. Proinsulin is a long protein that does not have any function in cells until there is proteolytic cleavage at these two specific positions. The amino acids in between get lost and degraded and recycled. And now the mature active form of insulin looks something like this. Before that proteolytic cleavage, that insulin or proinsulin would have done nothing to cells. This active form of insulin can do what insulin does. So proteolytic activation is one mechanism. Another one that is pretty common is myristoylation or palmitoylation. What that is a series of chemical reactions by which long fatty acids are chemically attached to proteins. And why that matters is because that provides these proteins with a chemical anchor to cell membranes, right? So here's a protein, totally cartoonish, generic protein, and now the same protein with these fatty acids chemically attached to them. These fatty acids don't like water environments very much. They don't like aqueous environments very much. They prefer very non-polar environments. Where will you find those environments? in the lipid bilayer of cell membranes. So these proteins will anchor themselves through those fatty acid tails to lipid bilayers. It could be the cell membrane, it could be the nuclear membrane, it could be the endoplasmic reticulum membrane, whatever biological membrane that is in a cell. And that places these proteins where they need to go to do their job. So it's not like this protein is totally non-functional before it got palmitoylated or myristoylated, but it may not be able to do its job because it's not in the right place, which is right next to a cell membrane. After palmitoylation and myristoylation, it can go there, start doing its job. Then there is phosphorylations and methylations, acetylations, a whole bunch of chemical modifications of amino acids in proteins. And what happens is, let's use this amino acid here as an example. So there is an amino acid here. We'll call it amino acid 33. That amino acid 33 can be one of three: tyrosine, serine, or threonine. The reason it can be one of those three is because all of those have an OH group hanging out on their side, right? And that OH group is where typically these things get attached to, right? Actually, methylations and acetylations, like we discussed with histones, happen in lysines and arginines. The point is that there is some amino acids with certain chemical structures, certain chemical features that will make them modifiable chemically by these chemical groups that get attached to them. So let's say a phosphorylation, like we were discussing before. So now a phosphate group is attached to that amino acid 33. Chemically, a phosphate group is a pretty strong thing because it has a bunch of negative charges. That's why DNA is negative, because of all the phosphate groups in it. So a phosphate group now attached to a protein changes the environment of this amino acid, or the neighborhood of the environment of this amino acid, quite drastically. Now that there is a strong negative charge around it, the rest of the protein, the neighboring amino acids, will have to react somehow. If there are positive charges nearby, they will come to it. They love it. If there are negative charges, now they're going to try and get as far away as they can from it. Either way, there's likely to be a change in the shape of this protein, and that is what turns on or off a protein after it has been translated.